How are proteins different from the random sequences from which they evolved, and how do they change with age?
Okay. A big question, evolution study a lot of genes, where do they come from? There is an old story that we all know about where new genes come from. They come from old genes. A gene duplicates, it diverges, one gene becomes two genes, one of these genes can be called old, one can be called new if you like. So there is an obvious question behind this answer, which is where did the old genes come from? And the answer obviously is that they came from older genes. Okay, so what we have clearly is some kind of chicken and egg problem which you have to resolve one way, and there are two ways of resolving it. And one is a sort of a Big Bang theory that all the protein folds, all the gene families that ever in existence were born in some primordial soup when everything was different, and nothing like that could ever happen again today. And I'm a member of the other camp, and I'm part of the continuous creation camp. And under continuous creation, new genes are born de novo, spontaneously, out of junk DNA on an ongoing basis throughout the history of life. You know, it's not as common a process as gene duplication and divergence, but it's common enough to matter and to give rise to new gene families. So what we have here is, I'll walk you through this, but ultimately is a finding that gene age matters. So on the x-axis, we have the measure of age using a technique called phylostratigraphy. So basically, we take every gene out of mouse and we blast P it against absolutely everything, and you say, where can you find it? And if you can only find it in mouse, it's a mouse-specific thing, and it goes here. If you can, if you can find it in mouse and rat, then it goes here. If you can find it in mouse and rodents, other rodents, it goes here, and so on. And each of this you sort of assign an age. What is going on? You assign an age of how long it's been since every gene family was born. I should also mention that we did something surprisingly novel here, which is we controlled for gene family identity. We did not consider the gene to be an independent statistical data point, but that phylogenetically confounded with other genes in the same gene family. I couldn't find anyone who'd done that before, which seems to be a big deal for the problem for the studying lots of different genes. Anyway, that's the x-axis. On the y-axis, we have, if you were here for Luke's talk, intrinsic structural disorder. If you weren't, this is being floppy, this is being highly ordered, this is hydrophilic, this is hydrophobic. You can calculate it using a program called IEPREP. So as you can see, young genes are more disordered than old genes, and there had been dispute about this before in the literature, but this is a nice clean result, and you know, it's been settling down for about 1 billion years. Okay. So the next, I should also say that this field of phylostratigraphy has been what you could call intensely questioned over an issue called homology detection bias. So you could ask, is it really true that these young genes are sort of causally responsible for high disorder? Or if you're disordered, you have a high evolutionary rate. If you have a high evolutionary rate, it's harder to find the homologs, and so you're more likely to be classified as a young gene. So instead of youngness causing disorder, maybe disorder causes apparent youngness. And here is the same data, but in a linear mixed model in which we corrected for DNDS, and you can see that the results are essentially identical. In any case, it wasn't very plausible because the signal was all in very young phylostrata, which even with high rapidly evolving genes, it's not that hard to spot homology. That becomes more of an issue back here in this deep time where we can't see anything anyway. Okay, so there's some theory here about how gene birth happens. A lot of people found de novo gene birth surprising. As if you just translate a bunch of random amino acids, it's going to be highly toxic, it's going to form aggregates, it's going to form amyloid, it's going to really stuff up the cell. And so there's been a few theories about why this can be done. And one of these, I'm showing you the paper just to show that it was in Nature, says that there is a continuum from sort of junk to something called proto-genes, you know, to young genes to old genes, and so it somehow happens in some continuous transition. And if that were true, what we would expect if we went out and had a look at junk DNA and we pretended that the junk DNA coded proteins and we plug that into IUPRED as a control that surprisingly nobody's ever done, we would expect to see the junk DNA come here as part of the continuum. However, that's not my prediction. My prediction was for a theory called pre-adaptation, which means the first thing, most important thing in being born is to first do no harm. And things like, you know, these toxicity and amyloid formation and so on all do harm. And the average intergenic DNA will do harm, and there are some properties that make harm less likely. And disorder is a property that makes harm less likely. Oops, I just tripped. I don't know why it's advancing itself. And so having that facilitates gene birth. And now you've already seen the punchline that the data we found strongly supports the pre-adaptation theory and completely falsifies the continuum theory. Okay, so what is responsible for this higher ISD on a biochemical level? And so it seems that mostly these yellow here are the same amino acids, and we just scrambled them with their order. And you can see the yellow is very similar to the red. So the pattern is primarily driven by the amino acid composition, but not exclusively. If you look with a paired analysis, not this sort of confidence interval that makes everything look non-significant, if you look in a paired analysis, the red is statistically above the yellow throughout the young genes. So most of it is amino acid composition, but young genes have another level of, at the sequence level that's making them more disordered. Old genes are not. So that's another independent line of evidence that something is favouring high disorder, specifically in young genes. So maybe this is not really about disorder. Disorder could be just a sign that amino acids are hydrophilic, that, you know, there are many things that correlate. And here we've got some junk DNA sequences translated, and you can see a very high correlation between, if we use a program called Tango to say how prone they are to aggregate, and you also look at their intrinsic disorder, you see the two are connected. So maybe the causal agent is aggregation, the causal agent is not intrinsic disorder. So we repeated the same thing using Tango and aggregation propensity, and we found the same result, not quite as dramatic, that young genes also tend to have a lower propensity to aggregate than old genes do, and again, in support of the pre-adaptation hypothesis. But we did see a really weird result when we scrambled them. We expected genes to be less aggregation prone than scrambled version, but the young genes, that's true for the very ancient genes, but young genes turned out to be more aggregation prone than their amino acid composition would suggest, which is really, really weird. So we had to think about this and what it could be, and thought, well, maybe it's because there's sort of some kind of clustering, that as a young gene, highly disordered, to have any hope of any sane structure whatsoever, you need a run of hydrophobic amino acids in a row in order to form some kind of fold at all, and that those runs of hydrophobic amino acids make aggregation more likely as well, on average, statistically. And then old genes don't need that anymore, because they know how to fold. So we looked at dispersion. We used a method called block technique that basically divides things up into blocks. We use blocks, and then compares the mean that you get in each of the blocks to the variance among the blocks, and get something sort of like a dispersion coefficient to measure how clumped together the hydrophobic amino acids are. And we get one of the strongest results we've seen so far on gene age. So this is clumping here, is what it should say. I wonder if it's the laser pointer that advances it. A Poisson line here is no clumping at all. And here old genes are over-dispersed. So they tend to alternate between hydrophobic and hydrophilic. And remember, most genes are old. You know, this is you know, you can see by the error bars, this is where a huge proportion of all gene families are. And this is actually what biochemists believe to be true. And I told a biochemist my theory, he said that would never work because he knew that actual real genes were over-dispersed, you know, were anti-clumped, and so that couldn't possibly be explained by results. So what we found is no, only old genes are anti-clumped. New genes are highly clustered, that the hydrophobic residues are close to each other along the length. And this trend goes back an even longer time. You know, we are talking extraordinarily deep timescales. So I think it's really interesting. Biochemists know that the protein folding problem is hard, but just to give you an idea of how hard it is, it takes evolution billions of years to solve it. That's hard. You know, this is directional evolution going on for billions of years. So, to sum up the story, we have strongly deleterious aggregation is a constant threat. There's a lot of lines of evidence that suggest that. The easiest way to avoid it and to be a happy protein is to be hydrophilic, which means that you have a goodly amount of intrinsic structural disorder as a protection. Young genes need to take a line of path of least resistance. Young genes need to go for this method, and then after that they have to evolve by, you know, descent with modification, and then they can only tinker after that to slowly become more ordered and fix the folding problem. But at the moment of birth, they are highly skewed towards hydrophilic amino acids and lots of disorder. If they're going to fold at all, then they need runs of hydrophobic residues within an otherwise disordered sequence, which runs an intrinsic risk of aggregation, but presumably the specific ones that happen in the real protein. Okay, and not the ones that were, you know, we only have theoretically predicted aggregation propensity. The real aggregation propensity might be slightly different, but there is in any case a danger there. It takes literally billions of years of evolution to switch these things up and actually get rid of these runs and come up with sort of a presumably more optimal solution that the very oldest genes have, which is to have over-dispersion of the hydrophobic residue, but you need billions of years of evolution to get there because it's that hard. And then you can tolerate much more hydrophobicity. Once you've got a really, really good fold, then the fold is the best protection of all against aggregation. Okay, and I would like to thank the people who actually did the work, which wasn't me, and the funding bodies, of course, and this is completely gratuitous.
First question there.
So I think it's also been shown by phylostratigraphy that new genes tend to be expressed more lowly than old genes. Is that correct?
I believe so, yes.
So is there evidence that at the relatively low expression levels that they're generally being expressed at, you have enough of a protein that aggregates are a problem?
Well, it certainly looks like aggregation is a problem in that the properties are very non-random and highly aggregation avoided.
You mean just based on the plots?
That's
is there
right.
like experimental evidence to that effect?
No. I mean, there's very little done. There's much more work done on old genes. When you look at the experimental literature, you get a massive skew towards the specific study of old genes.
Sure. Thanks.
Okay.
So you controlled for DNDS in that one thing you had. So the search, presumably the homology search was with protein sequences, right? And
yes.
I do worry that DNDS over that range is sort of not very confident.
Oh, sorry. Yes, the homology search was with protein sequences. The DNDS was all mouse versus rat because that's the only thing that was consistent. Yeah. And then when there was any ambiguity, we took the fastest one. I think that's all I'm allowed to say.
Okay.
Because I have to give